So good morning and good evening, ladies and gentlemen. Welcome to the MSQ webinar. So we're going to have this webinar every Thursday, Beijing time 9 p.m. to 10 p.m., New York time 8 a.m. to 9 a.m. Hello again, my name is John Ma, and I'm a senior associate at MSQ Ventures based in New York City. So basically, MSQ Ventures is a cross-border advisory firm focusing on healthcare products and China. So basically, we bring our Western biotech companies, clients back to China for fundraising and also from GV and all other related products. So today is it's a great pleasure for us to have two experts from Leidos joining us to talk about their pioneering work in immunovirology. So I'd like to hear from my co-host, Dr. Shang Shi, my colleague at MSQ. Shang.
Yeah. Hi, everyone. Sean here. I'm Beijing venture partner at MSQ. I'm based in Beijing. Before joining MSQ, I was healthcare investor in China, doing some PE and VC investment in the China healthcare industries. And I've been with MSQ for more than half a year. And just like John said, we are helping the companies to do some cooperation in China. And today we're talking about the subject on the virus pandemic, the futures and the current situation.
So I'm going to go ahead and let our guest speakers introduce themselves. So first would be Dr. Gabriel Gutierrez. Please, Gabe, please go ahead.
Hi, John. Thank you. Thank you, Sean, and to everyone, good morning, good afternoon, and good evening, wherever you might be. My name is Gabriel Gutierrez. I'm the scientific director for the Explorations in Global Health Division within the Leidos Health Group. I've been doing cancer research since my postdoc days at medical school with Phil Hines. And after doing my postdoc in cancer research, I moved over to Leidos, where I took over as a principal investigator on a malaria vaccine development contract, which may seem very unique to move from cancer to vaccine development. But at the time, it was a transition that we were looking for novel types of vaccine products, prototypes, against a pathogen that looked to be sort of avoiding of the immune system. And at the time, we were just learning about how cancer was starting to avoid the immune system by using the checkpoint inhibitors, for instance. And so we actually sort of fit into that. And that's how we sort of evolved from our position as vaccine developers and into cancer, and then back into hopefully talk a little bit about how we're studying viruses and other pathogens. And so my PhD was in genetics, background in cancer, but a lot of work in infectious diseases and particularly vaccines. Tim?
Thank you, Gabe. Tim, please.
Thank you, John. Thank you, Gabe. Hi, my name is Tim Phares. I'm the head of immunology within ExGLOH. My background is immunology and infectious diseases, where I did most of my graduate and postdoc work working on neurotropic virus infections, so a lot of neuroimmunology. After that, I went to the Walter Reed Institute of Research, where I worked on malaria vaccine development. And then in 2016, I left Walter Reed, and that's when I joined Leidos as an infectious disease scientist, where I have been working on the malaria contract that we hold with USAID, as well as working on the peptide that you'll hear about today. Thank you, John.
Okay, thank you, Tim. So let me explain a little bit today's agenda. So basically, we're going to go through the presentation for about 20 minutes, 20 to 25 minutes. Then I'm going to have a, about 30 minutes for Q&A and discussions. And so I'm going to stop sharing my screen, and Gabe, you can run the slides from your side, okay? Go ahead, Gabe.
Yep. Give me 1 second as I load them. John, can you see my screen?
Yes, very well.
Okay.
That's good.
Great. So I think I alluded to this a little bit when I spoke about myself and what we're trying to accomplish here. But very broadly, let me just introduce Leidos and the explorations in global health, just to give you context of where we live inside of our organization. Leidos is a large, primarily US government contractor focused on defense intelligence, aerospace and engineering. And that's about 35,000 employees, $11 billion in revenue a year. We live within a health group inside of Leidos. That health group is about a $2 billion organization and dedicated to public health as well as promoting health within our VA system, our military health, and the hospital systems across the nation and across the world. We are a dedicated drug development group. There's only about 6 or 7 of us inside this small group. And we were born out of a lot of the contracts that we did to support the NIH, USAID, and other US government agencies. I was the principal investigator on a $76 million malaria vaccine development contract. And we then took over a $25 million 5-year contract with USAID to develop those malaria vaccines. We've worked on a lot of other diseases, but I've been the principal investigator on those 2 contracts for several years now. And that's basically where we came up with this idea about 3-4 years ago that infectious diseases like malaria and other pathogens are hiding in plain sight of our immune system, not unlike cancer, in the sense that over 30 years of trying to make traditional vaccines against malaria have failed. And one of the big reasons we saw that, or one of the big questions we wanted to sort of answer and get back to, was how are they doing this? How are they sort of frustrating our immune system? Well, a big inspiration came about 3-4 years ago when all the data from Keytruda and the BMS were starting to sort of come up, where we were using the immune systems, our own immune systems, the host immune systems, to fight cancer itself. In fact, just this morning I read an article that said that the only 2 ways to treat a viral infection was to either attack the virus and its replication machinery once it gets into the cell, or use a monoclonal antibody to prevent it from getting into the cell. And what we're trying to propose is that there's another way, the way that's the same way that sort of the cancer revolution is doing it, and that's using immunotherapy, using the immune system, the host's own immune system to attack those pathogens. So in order to address that and to sort of begin to create molecules that would address that question, we developed what we call the Microtide platform. What we did very straightforward was to look for peptides. And the reason we chose to look for peptides was that we understood one very basic fact about the microbiome and the microbiology, is that the microbes speak, or their grammar, their language is peptide. All the antibiotics, they fight war with each other. These are peptide derived. They punch holes in each other's cell walls using these peptides. And we've subsequently used those as antibiotics. Microbes also quorum sense, and they do a lot of other activities. And that language, again, is the peptide. So we use that as a sort of a bio-inspiration. So to use novel chemistries, we use the microbial database. So we took some random peptides, but we sort of leaped into the microbial databases to see if there are some peptide scaffolds that we could use from there. And once we started getting a lot of success in looking for those peptides and deriving new peptides that hit these receptors like PD-1, we began to optimize them, use more AI tools to do that. So that led us to sort of several lead candidates that I'll show some data on that had activity against PD-1, as well as PD-1 and CTLA-4. So there's a wonderful thing, I think, about these chemistries, bio-inspired chemistries, is unlike traditional sort of US pharma, we're not, the microbes aren't hyper-focused to a target. They're interested in an outcome. After all, they want to live on us. 50% of us or more is microbiome, and they want to maintain a balance or a homeostasis on us. So they're not interested in a single target. They're interested in maintaining homeostasis of our entire immune system. That means there's an opportunity for polypharmacology, meaning we're probably going to hit more than one of a family of receptors. In the case of PD-1, CTLA-4, which belong to the CD28 IG superfamily, we indeed found that our lead candidate, which we call LD, hits several of these CD28 families. And so what we'll show today is that not only are these going to be effective in cancer, but they have a utility in global health. And in particular, we're hoping to show that with viruses, particularly the COVID-19. So that's just a little bit of background on what we have and what we've done. And I just kind of want to sort of talk a little bit about what we're trying to accomplish by hitting the host immune system. This was a paper written back in 2001, where it was clearly understood, and the quote is, "Viruses have studied immunology for over 1 million years of coevolution with their hosts. During this ongoing education, they have developed countless mechanisms to escape from the host immune system." And I think that's very telling, right? Evolution has taught us that, there is a battle between the viruses and I think what we've really understood, and this is where the crux of what we're trying to accomplish, is that how they do this, to answer that question that I told you we were trying to do in the vaccine setting, is how are they escaping? And to understand that, you have to look at the CD8 T cells and how they respond to viruses, and how you can predict the outcome of whether you're going to basically survive, or are you going to succumb to this virus? So if you look at the right here and the top panel A, and again, this is taken out of that paper from Lucas et al. What they show is that if quickly after you get a viral infection and you start to begin that viral load, to see that viral load in the serum, CD8 kick up, spike in parallel with the viral load, and then quickly those CD8 cells control the virus, and then you'll have a favorable outcome. Then there's also another case where what we call more a chronic or a persistence, right? Where you're going to get a very high viral load very early on, and then when the T cells kick in, this is where there's the opportunity for the viruses to frustrate the immune system by creating an exhaustive state to those CD8 cells. And if that sounds very familiar to cancer, I think it should, because when we talk about exhaustion in cancer cells, we talk about tolerance to those tumors. And in this case, we're talking about tolerance to those parasites, persistence in the host cell. And that obviously causes a whole lot of issue with viremia and those negative outcomes. So, that's it's a persistence, and that can be a very dangerous case, right? Where we're overcome with viremia and all the secondary effects to the other parts of the other organs, and something that resembles sepsis and eventually sort of succumb to the disease. So in the intermediate case, in panel C, this is where we see sort of a persistence of the virus, where there's a slow and steady intake of the uptake of the virus. And then there's sort of this increase of CD8. And I think there's an eventual clearance there. There's sort of a battle between the immune system and the virus. But eventually, this is in this case, you overcome the viral persistence, and eventually the immune system wins. So this is sort of the background in terms of what we're trying to achieve, or at least what we're trying to address. This is the CD8 compartment is really where the battle lies, and this is what we're trying to achieve with these checkpoint inhibitors. And so this just elaborates a little bit more on what we talked about earlier, where the COVID leads to this respiratory, the acute respiratory, or ARI, not unlike influenza, RSV, and other viruses like adenoviruses. These are associated with CD8 T cells that exhibit that immune or impaired function. There is a slight difference between when we say impairment and exhaustion. Impairment typically connotates something more in the case of an acute dysfunction, where exhaustion has more of a long-term or persistent phenotype of the T cells. So T cell impairment usually happens within this, after 7 days of seeing the viruses. And what you'll see is that a PD-1 in particular is upregulated. And that's where you see that exhaustion to start to kick in. So when we think about PD-1, we think about this as a checkpoint inhibitor, classically as sort of waking up the immune system or causing this, immune reaction, right? But I think there's a lot of people out there who really think of it as more of a rheostat. And in fact, the whole immune system is not necessarily as a barrier between you and the outside world. Immune system has this connotation of a political connotation of them and us, where in reality, like I mentioned earlier, the microbiome is part of us, right? And what we need to do is establish homeostasis. And so when you think of the immune system in PD-1, you want it to be set somewhere where it is comfortable for them and for us, where we both can tolerate each other. And so this PD-1 creates that balancing act, right? And that's what we're trying to do, is trying to get in there and identify, how the microbiome is doing it, as opposed to trying to create something that's unnatural, like a monoclonal antibody that shuts it off completely, and then causes a very sort of strong, I think maybe a too strong a reaction, which can whip the immune system or that rheostat in the wrong direction. And in fact, in the case of blockading of the PD-L1 or PD-1, it increases. We know that when we hit that rheostat and we drive it to something that's going to be drive the immune system to try to wake it up in a way, it does in fact revive those CD8 T cells to recognize viruses such as the HMPV or the influenza, and it drives clearance of those viruses from the host. So I just put in some statistics that I'm sure most of you guys know better than me. I'm really just a dumb molecular biologist in this case, and what you probably understand much better than me is that there's a tremendous burden in influenza, and I'm just using sort of that as an analogy to a potential pandemic and where we sit today with the COVID-19. And there's not much here that I can say other than using this as sort of analogy or casuistry to show that it shows a lot of the same signatures, it turns out, whether you get that septicemia, the viremia, and really the complications, where we get shutdown of internal organs, and which is eventually overtake the patient. And a lot of these signatures are very similar. And so we can expect to try to use that as sort of an analogy or jumping point, and how we might be able to treat this or understand this. So this gets to where we sort of come in with with our concept or our hypothesis, really, because what I'll show you is a lot of data on infectious diseases, but not necessarily the viruses, and that's kind of where we want to sort of engage with the community, is to understand how we can apply these to a disease like or a viral disease like COVID-19. What's clearly been shown in the case of influenza, and so we'll stay there as sort of a case of casuistry here, is that PD-1, when blocked, and we're talking about blocking it with the monoclonal antibodies, we're talking about Keytruda and that class of molecule, it does improve T cell function and clearance of a highly pathological influenza A virus. This is done in mice, by the way. And that is directly correlated with that CD8 T cell reanimation. So we know there's very good data that shows that T cells, the blocking of the PD-1 receptor has a really good chance of waking up the immune system. And we could talk about the molecular mode of action of that activity, but suffice it to say, it really does point to CD8s as being sort of very crucial to that response, right? That exhausted or that impaired state of those immune cells. And once you wake them up, or you bring them back online from that exhausted phenotype back to an active phenotype, they now are able to recognize that foreign or that pathological immune signature and antigens, and then go after it and attack it. However, we do know that T cell functions have detrimental effects in the lungs. There are rare and serious side effects. That's primarily due to inflammation in the lung, right? The pneumonitis, and these other types of, these other sort of overreactions, right? And I think I alluded to that earlier by saying, when you take a monoclonal antibody against the PD-1, it circulates for weeks, if not months, in your in the serum, and that kind of hyperactivation on that PD-1 receptor is eventually going to cause that autoimmune activation on those epithelial tissues, right? And not just targeting those exhausted immune cells. And I think the idea is to wake up those exhausted immune cells, have them recognize the pathogen, and then sort of start that cascade of killing them. But when you take a monoclonal antibody, which is going to be persistently present in the host for weeks, that's where you're going to be able to cause that pneumonitis and these other auto-inflammatory diseases. And so that's where we came in. I think I spoke about that earlier, that what our sort of vision was, can we find something that was going to be gentler, that's going to be mimic what nature has already done, which is derive something from the microbiome that's peptide based, which is already that language that the microbes speak to hit these checkpoint inhibitors, which we call LDO1, which we found and we know that hits the PD-1 receptor. We call ours LDO1 or the LD class. And we showed that it has potent activity, similar to those monoclonal antibodies. But that potency is not derived from its ability to sit there and hang out for weeks or months, but its ability to hit a broader range of those CD28 checkpoint family, namely PD-1 and CTLA-4. And I think we have data that it hits other members of that CD28 family, but it does so in a pulsatile or a more gentle fashion. So you cover a broader waterfront of these receptors, but you do it so that you don't, hyperactivate the immune system. And that's partly due because we have a 30 to 60 minute half-life, which is in a peptide relative to other peptides, like let's say insulin, which has 6 to 10 minutes half-life and a very potent activity, we still have a very long half-life relative to, like, let's say insulin. But we cover a broader swath of maybe somewhat redundant checkpoint receptors, like the CD28 family, and we have just as good an outcome. Therefore, and this is where we come to the crux of our story or our hypothesis, that we want to be able to use these LD class of peptides and mind you, they're synthetically manufactured. They are about 18 to 20 mers, 20 amino acids, linear. There's nothing unusual about our peptides. So they're rapidly deployed. Within 30 days, I assume you can make enough of an enormous batch, especially at the capacity that maybe China has to manufacture. And we can use this as an antiviral therapeutic that raises the immune response, recognizes the antigen, and prevents that some of those secondary effects. And it's important to note that while, again, I mentioned that we haven't tested these in either the corona or flu just yet as therapeutics, we have some data in sepsis, which I think correlates nicely with the viremia and the septicemia that's sort of correlated with the morbidity around COVID and other influenza and other acute respiratory infections, viral infections. Okay, and so I think I just want to sort of emphasize this point, because obviously there are a lot of checkpoint inhibitors out there, right? I know that China has its own pharma industry going after some of these checkpoint inhibitors. Beijing, the big pharma biotech giant there in China, has its own PD-1 inhibitor, monoclonal antibody. But again, I don't think that the monoclonals are going to be the right tool for an infectious disease for a lot of reasons. And I think primarily it's that immune-related toxicity. You're not going to want to treat that inflammatory response with something that can potentially create more of an inflammatory response. That's where the peptides, I think, show their mettle or have their worth. There's a shorter half-life, right? The local delivery limits the systemic toxicities, so we can deliver them in the lung, for instance. They're small. Or if you want to deliver them systemically, we know that they have broad activity system-wide, host-wide, when we deliver them sub-Q, for instance. So they're easily manufacturable. They're low cost. I think, China particularly has an edge on a lot of that large capacity, especially in the peptide space. There's a lot of interest there. And then the flexibility, right? So if we wanted to put them in, for instance, genetically engineer it into a viral vector to get it quicker into the lungs or spread it quicker, like we know that we can express it in a DNA construct delivered in an MVA vector, for instance. And so there's broad utility as well. I think we understand everything from cancer to infectious disease, even as a vaccine, potentially as a vaccine immunomodulator. We show efficacy in those regards. We're confident of what we do, and I think we're very excited to be sharing some of the data that I will be sharing in a moment. But just very broadly, I think what we want to talk about today is our lead candidate. This is that dual checkpoint inhibitor, the one that hits not just PD-1, but its cousin, CTLA-4, of the same CD28 family member. And I also sort of alluded to that briefly, that not just PD-1, CTLA-4, but maybe other members of that CD28 family. It's a small peptide, about 20 amino acids. It's derived from a microbe, a bacillus. I think it's something that I think is very familiar to a lot of food people as well, because this is not a microbiome, and I should say that this is microbial, and it's a food microbe primarily, and not necessarily microbiome. So that I just wanted to clarify that before we move forward. We know that it has polypharmacological activity, as I just emphasized earlier. This has been computationally optimized, so we'll show a little bit of data on that, and as we go through the deck. And we know it has activity both in because of its targeting of those checkpoint receptors. We see an increase in proliferation. We enhance that T cell activity, and we can show that in mice. And it has, and again, in those mice, we show that it has improved outcomes, both for therapeutically, but as well as an immunomodulator in combination with a vaccine. So I just want to just say that, you know, we have a lot of data on this. We have a lot of confidence in this. We have the first generation of these peptides sort of ready. And we'll show some of those data that it's shown to be safe, easily manufacturable, and efficacious, and ready to be deployed. And I think we just need some you know, proof of principle really at this stage. But I want to turn it over to my colleague, our head of immunology, Tim Phares, to sort of walk through some of our the data that we have with regard to this peptide.
Thanks, Gabe. So what you're looking at here on this slide is actually a cell-based functional assay that we typically run after we identify our peptide hits. So we're looking at here both cell-based assays for PD-1 and CTLA-4. This is a assay that has been developed at DiscoverX, which is now owned by Eurofins. And very briefly to tell you, in the case of, let's say, the PD-1, PD-L1 interaction, you have a cell that overexpresses PD-1. You have another cell that expresses PD-L1. When PD-L1 binds to PD-1, this leads to the recruitment of the phosphorylation of the PD-1 cytoplasmic tail, which leads to the recruitment of SHP1. And attached to this SHP1 is an enzyme that allows for, in the presence of the substrate, for chemiluminescence. So if you have engagement of PD-L1 with PD-1, this allows for the cell luminescence. So if you now add an antibody, or in our case, our LD peptides, you block that engagement of the receptor ligand, and therefore you will decrease that chemiluminescence. So shown on the right-hand panel is a series of our LD peptides, LDL1, LD10, and LD16. And you can see that both in the PD-1 assay as well as the CTLA-4 assay, that at the two different concentrations, we can inhibit that interaction of PD-1, PD-L1, which again, the readout here is a decrease in that chemiluminescence. Also shown here is LD12. You'll see on the very far right. This is actually a peptide that has a single amino acid change compared to the other peptides. And you can see that we can completely ablate the inhibition of the LD peptide when we make this single amino acid change. Next slide, please.
Trying there, Tim. It looks like I'm a little bit frozen up. Hold on.
There we go. So here we're showing the efficacy of our peptides with human PBMC cells. So what you're looking at here on the left-hand panel is a stimulation of PBMCs with the superantigen SEB, where we're looking at the production of interferon gamma, measured here by looking at supernatant levels. And you can see that with the SEB, you get a certain level of interferon gamma. If you add to that culture Keytruda, you can see that you can increase that amount of interferon gamma. And then you can also see here that with our LDL1 peptide, you can see that we can do as well or even slightly better in the production of interferon gamma when we add the LD peptide to PBMC assay. On the right-hand side is a very similar assay. However, what we've done here is specifically looking at virus-specific CD8 T cells. So here we've stimulated or done a recall assay with the PBMCs for CMV specific epitopes. And shown here are two of our lead human candidates. That would be LD10DA and LD16DA. And you can see that if you add, again, on the far right is Keytruda. So you can see that you get a certain increase in the percentages of tetramer positive CD8 T cells, again, identified by flow cytometry relative to the IgG4. And you can see with the LD10 that we do very similar to the Keytruda expansion of these CD8 T cells. And we see even a further increase in the groups that were treated, or the PBMCs that were treated with the LD16DA. Again, so this is just showing you efficacy in the context of human PBMCs. So also, Gabe really stressed this point, and this is something that we really focused our attention on to be able to apply these peptides not only in the cancer field, but particularly in the infectious disease, either therapeutically or in the vaccine world, is that we needed these peptides to be safe. So some of the studies that we've done so far in mice, we've done a non-GLP tox study where we dosed with several different doses. And what we've seen so far is that there's been no abnormal either weight loss or injection site irritation or behavior or anything within the necropsy of those animals. And then also on the top right is just showing you that, again, injection of these peptides don't cause any weight loss in the animals. On the bottom panel, or half of this slide, this is showing you that if you do inject these peptides, this was a single dose IV of 200 micrograms, that you don't non-specifically create any kind of cytokine storm or adverse event in those mice when these peptides are injected. Also, Gabe had mentioned this as well. We've done some PK studies as well as to look at the immunogenicity of the peptides. So on the top left-hand panel, this is just looking at some of the PK data for the LD peptides, as well as our LG peptides, which is peptides that we've developed against LAG3, which we'd be happy to discuss further in the discussion section. But focusing on the LD peptides, you can see that their circulation stability is anywhere from 30 to almost 2 hours, depending on the peptide. And also, I should mention that we do have the tissues. We haven't looked at whether perhaps these peptides are being sequestered within the tissues during these PK studies. On the far top right, this is just showing you that we haven't found any reactivity to the peptides as far as a T cell response. So this was in a TV model that we actually looked at, where we dosed the animals 10 times over 20 days. And what we did was to take out the CD4 or CD8 T cells and then incubate those cells with the peptides that we had injected to the animals. That would be the LDL1 or LDL2 that you see there. And you can see that we were unable to detect any, shown here is the CD8 T cells producing interferon gamma, but this is also the case for the CD4 T cells. So suggesting that we don't mount a T cell response to these peptides. In the bottom half of the panel, this is showing we wanted to look at whether we are inducing an antibody response. So what we did was to actually take serum from mice that we treated in a vaccine model, where the peptides were injected 4 times after 3 immunizations. So that's a total of 12 injections of 200 micrograms of the peptides. And what we did see is that depending on the mouse strain, we did see some level of antibodies raised against the LD peptides. You can see in the far left of the graph below that in the BALB-C, there was really no response. but in the C57 black 6 mice, we did see a fairly good response. But again, I want to point out here that these mice were pretty much very heavily dosed with the peptides, where in the case of many of the studies that we're doing now, we're only giving a single or a very limited amount of doses during those studies. So as Gabe had alluded to, we haven't necessarily looked at treating or administering these LD peptides in the context of viral infections. But however, we have looked at both sepsis and malaria. And both sepsis and malaria very similarly show either T-cell impairment or T-cell exhaustion, particularly the case in sepsis, where it's been termed immune paralysis, where, as you know, in sepsis, many of the individuals who do survive sepsis actually then either succumb to a secondary infection because their immune system, again, is either immune paralyzed or immune suppressed. So if we look at the sepsis model, again, this is called the cecal ligation puncture model. Very simply, the mice are opened up, their cecum is punctured, and then allowing the microbiome to spill into the peritoneal. They're sewed back up, and then they have a peritonitis. And what we were able to show in the, you can see in the top left-hand panel, is that with the treatment of our LDL1 peptide, that we can increase the survival from about 50% to about 70% or 80% in some studies relative to the vehicle control. And you can see that that was slightly above the anti-PD-1. You can see that when we went ahead and looked at the bacterial burden, you can see that with the treatment of our LDL1 peptide, we had a significant decrease in the bacterial burden in the peritoneal cavity. And also, what we were able to show is that this increase in survival and decrease in bacterial burden was correlated with an increase in phagocytic activity of the peritoneal macrophages, simply by looking at the ability of these peritoneal macrophages to digest or phagocytize FITC labeled E. coli. Not shown here, but we also know that we can enhance the T-cell activity in these septic mice by looking at their splenocytes, where we have an increase in interferon gamma either by ELISpots or ELISA when the mice are treated with LDL1 compared to the vehicle control group. Now, similarly in malaria, again, a parasite which really manipulates the immune system and causes immune dysfunction. In this malaria-specific model, this is a lethal malaria model, where you can see that mice that are infected with malaria die within 2 weeks of the infection. However, if you treat those animals with either our LD peptide or an anti-PD-1 mAb, you can rescue about 40 to 50% of those mice, and we've seen anywhere as high as 60 to 70% with our LDL peptide. Now, this increase in survival, just to show you that this does lead to a clearance of the parasite from the mice. And then what we also wanted to do was see whether, now that we had treated these mice with either the LD peptide or the anti-PD-1 mAb, whether we had induced a long-lived protective immune response. So what we did was to take those mice that survived, and then 100 days after the primary infection to then infect them with now, again, a lethal dose of the same parasite. And you can again see that those mice that are naive quickly die from the infection, but those mice that were given either the LD peptide or the anti-PD-1 mAb survived, showing that in the context of the malaria infection, essentially we were able to vaccinate these animals and generate a protective long memory response, which again was resultant of adding the LD peptide or also in the case of the anti-PD-1 mAb. And then just to follow up on what Gabe was saying is that we also think that modulating the checkpoint inhibitors during vaccination is also efficacious, as we can have an effect on the CD8 T cell response, as Rafi Ahmed showed a couple years back, and others have been showing that PD-1 is and other checkpoint inhibitors are quickly upregulated on T cells during differentiation. So if we go ahead and block this interaction, what we would hypothesize is that you're going to then allow for the T cell pool to expand, and therefore that increased expansion will lead to more of a memory pool population when you then encounter the antigen. So what we're looking at here is actually 3 different platforms or formats of malaria vaccines that we have studied. The first one is an irradiated malaria sporozoite vaccine. And you can see that with the addition of our LD peptide. Now, this is just a single administration of our LD peptide at the time of vaccination, that we can significantly increase the number of antigen-specific CD8 T cell responses measured here by ELISpot, and that's comparable to the anti-PD-1 mAb. The second or middle panel shows you a replication defective adenoviral vector vaccine that we use. Again, you can see that the addition of our LD peptide, again, a single dose at the time of vaccination can significantly increase the number of antigen-specific CD8 T cells. And then on the far left panel, this is where we actually used a recombinant protein, RH5, in combination with alum, which many of you may be familiar with. That alum doesn't produce a very good T cell response. It's more of an antibody response. And what we were able to show here is that if we added our LDL1 peptide, we could significantly increase the number of antigen-specific T cells. Just one thing to point out, that last bar in that graph with the addition of the LDL1 peptide, what we did here is that rather than giving the peptide after giving the peptide after each immunization, which there was 3, we decided to only give the LD peptide after the second and third immunization. And as you can see here, that resulted in a further increase in the generation of antigen-specific T cells, which really speaks to perhaps the timing of when we want to add checkpoint inhibitors, not only in the case of cancer, but perhaps also in vaccination, where perhaps allowing or delaying the dosing till the second immunization is more efficacious. Also, I just want to quickly point out all the data shown here. We've also tested this in a Pseudomonas whole cell vaccine. And what we've shown there is that when we give the peptides after, again, a single injection after 2 immunizations, we found that we were able to significantly reduce the amount of bacterial burdens in the lungs in those mice that were challenged. And in fact, what we saw was that in 2 of the 5 animals that we looked at, we had completely cleared the Pseudomonas from the lung, which the collaborators that we were working with had never seen that with the whole cell vaccine. And again, this is just a part of our team, I should say, our Vice President, Jim Panucci. We also have other members that are not listed here. And I guess we'll just start the follow-up questions and discussion.
All right, thanks a lot, Gabe and Tim, for a very fascinating talk. Actually, I think this is one of the first times that anybody has ever mentioned the this all new kind of like immunovirology concept. So please keep sharing your screen, and in case somebody has questions regarding some of the data points. Also to the audience, please, there's a raise your hand icon at the bottom of the screen. So if you have any questions, just press that button and enter your questions there. So I'll start with a couple of questions. We have about 10 minutes left, which should be enough for 5 questions also. So this is regarding the current COVID-19 epidemic. So basically, even though we haven't tried anything on either flu or a coronavirus mouse model. So basically, I think now people, we have thrown everything that we've got, from HIV drugs, malaria drugs, flu drugs, onto this this new coronavirus. And so far, I don't think anything has really proven to be effective. And also, people have been trying something focusing on the CSS, the cytokine storm. So basically, from IL-6 antibodies to even STAT inhibitors. So, Gabe, can you share with us such kind of approach, the difference between yours and also, some of the things that I just described?
Yeah. So what I know about, for instance, the STATs and the IL-6 from the case of the cytokine storm, it's something that has a lot of sort of prior use to use in sepsis, for instance. And I think that there's showed some limited success when you have a very early or acute, what they call acute sepsis. When we talk about something like sepsis or a viremia, it's a very complicated disease, right? There are several diseases in there, but initially there is an enormous amount of what the immune system is trying to accomplish very early on when it throws everything at the pathogen. It is highly immunogenic, right? But as I mentioned, it starts to then become exhausted. So when you try to do something like steroid use or IL-7 or STATs to try to cool that immune system, you're sort of, what you're getting wrong about that is that the immune system doesn't need to be cooled or sort of tampered down. It needs to be focused. At the moment it is exhausted, those CD8 T cells are there. They're just high expressing PD-1, for instance, and they're misdirecting fire, and that's misdirecting fire to things like your own autoimmune antigens, and so that's causing that shutdown of the secondary tissues, et cetera, right? So I think when we talk about what we're doing with checkpoints, it's not that we are trying to, raise an immune alarm. What we're trying to do is just lift exhaustion. So unlike the IL-6 or the STAT inhibitors or even the steroids, we're not trying to calm down the immune system. We're trying to redirect it. It's almost like bringing it back online. So a little bit different than that approach, very different from that approach. But I understand that it's still looking at the immune system as kind of the problem. And what I'll say is that's correct. It's just that you have to understand that the immune system is not that it's overactive. It's just that it's misdirected. It's exhausted.
Okay, that's very, very good. So basically, the keyword here is refocus. So you already have the weapons and you just want to refocus them onto the virus itself, not, your own tissues, right?
Exactly.
Okay. So that I think that's kind of like a paradigm shift, and also even becoming philosophical. So when you talk about, like single gene disease models in the early days, and also now we are into the immuno-oncology era and in systems biology, basically. And so it's very interesting to know to let you know that actually some of the traditional Chinese medicine has been working pretty well, pretty effective on last time when SARS was here, it saved a lot of people's lives, and now I think people are using this very for some very good results with this new coronavirus infected patients. So in your opinion, how does that connect to what you're doing here? I mean, the traditional Chinese medicine or the you know, medicines from India and or South American. So you know, is there a connection?
There is a connection. What I'll say is that when you have to sort of look back at first order principles when you think about what are you trying to achieve. In the case of you know, a pathogen that's evading or frustrating the immune system, you have to ask why. You can't just keep throwing, you know, traditional sort of, you know, I just have to combine this antigen, or in a vaccine case, this antigen with this adjuvant, do that for 30 years and get nothing, and then sort of keep trying to do the same thing. I mean, that's the definition of insanity. What you have to do is understand, how is it thwarting the immune system? Well, how is another question is, how are they living in balance with the immune system? How are we living in balance with our food? Food can be just as toxic as a microbe, right? We know that. More people are dying from diabetes and, you know, leaky gut syndrome that causes autoimmunity, et cetera, if we're not careful with our food. So there is a balance with our food. And of course, there is a balance with our microbiome. There's more of them than us living inside us. We have to strike a balance. And how are they doing it? Well, I mean, it becomes, with food, it becomes, there's a lot of issues, right? In terms of quantity, but also what kinds of food. So I think the observational biology of traditional Chinese medicine is spot on. I think the idea that microbiome is manipulating the immune system is spot on. Are we hitting, you know, are we, by targeting peptides as metabolites of that, is that the right thing to do? Well, we guessed on that. And I think that we may have guessed correctly. Are we in any way sort of onto something bigger? Maybe. We may have gotten lucky, and we may be just, but we certainly, if anything, just scratching the surface. But traditional medicines based on foods and, you know, our environmental microbes and you know, not just the food, but the microbes that live on the food are just as important as you know, basically we're saying. So I think that that's where we sort of intersect with that traditional medicine, is that you have to sort of go back to first order principles and use that observation as how are we maintaining homeostasis with our environment? Like I said before, the term immunity really is a bad term, right? It's a political term of, you know, we are exempt from. We are not exempt or different from our environment. We need to live in balance with it.
Okay, thanks a lot, Gabe. So